Gracias por la invitación. ¿Se escucha bien atrás? Sí? Okay. Well, I'm going to give this one in English due to our visitor. So this is something they did for me in Denmark, so I will do it now, even if I was the only one not speaking Danish. So all the slides are in Spanish, so if you have any questions, you can do them Spanish, English, it's the same, and you can stop me at any point. We'll talk about a little bit about the research I'm doing at the moment, which is the neuroimaging of cocaine addiction. And of course, I will talk about you just saw one of the images and some of the sequences, but I will make it a little bit brief, the clinical part, and try to talk more about the methods part. So it's an ongoing work at the moment. You heard about that. So in general, my basic research question is how does abuse and substance addiction affect the structure, function, and cerebral circuits, and what is the neurobiological substrate of that and the consequences. So it's a big question, so that's the umbrella question that I have. And well, for now I just use the MRI, but this is something I want to do in the near future. And we do also clinical evaluation and cognitive as well, and mainly humans, but I'm starting to work in rats and mice at the moment. We're going to start a project about that. And my idea is to try to bring the basic research to something more clinical or more translational, if that's possible. Basically, what I study is cocaine at the moment. That's the main question I have. It's about cocaine, and this is because you have to separate the other drugs. They have mostly the same effect, but it seems it is not exactly the same. So right now we're focusing on cocaine because it's the second most used in the world and also in Mexico. Even if you don't know it, yeah, there's the second most used. The first one is the cannabis, but it's the one that produces more problems. It's crack, and which is the rock type of cocaine, and then there's the inhaled cocaine. And the high is better in this one, so the high lasts more time, and with the crack is less. And this is the problem because it creates more addiction to the patients and apparently more cognitive and clinical deterioration. So the phenotype usually it's so the addict, not the user but the addict, has a lack of control of the consumption of the drug. They have problems in everyday life. Social problems as well. They develop tolerance and abstinence syndrome if they don't have the drug. So that's a huge problem that makes them take the drug again. And there is no cure in humans, at least that we know of. And it is interesting that in murine models, about 10% of the animals exposed to the auto administration of the drug actually produce the addictive phenotype. And it basically it's similar in humans, so that's in the epidemiology research we have seen that. So not everyone gets addicted, and this is quite interesting, which is that some people get addicted, at least in the murine model, some rats or mice get addicted. Some of the other ones just recreationally take it, and they're not addicted, at least in the formal model way. And then there's rats that actually don't get addicted to the drug. So it's quite interesting. And mainly the cocaine acts in the dopamine system and in these three types of systems. I will just go fast on this one because there's a lot to cover. And there's different models of how the addiction begins, or it keeps affecting the person, which is one of them is the theory about being a disease, a type of cycle that Nora Volkovs mentions here in this new paper she has. And it's basically you have the intoxication or the binge. So the first time you take it, everything changes, and then you have a withdrawal effect, which causes a negative effect related to areas of the brain. Yeah, and this then goes to preoccupation and anticipation, also related to these areas of the brain, and you take the drug again. So that's the basic cycle of addiction in all types of addiction, also alcohol. But it's what the areas are very similar in all types, but cocaine is a bit different than the other ones. There's a lot of clinical alterations. I won't go into it, but we have a lot of psychiatric problems like suicide risk, depression, etc. And of course, there's never only one substance. There's usually more than one substance. So research in this type of population is quite a challenge. And then you have the cognitive problems as well. Deficiencies in attention, inhibitory control, decision making, of course. They have decision making problems because they decide to take the drug and then take it again. So there's a lot of problems with this patient, but not as much as you would think, or as that's what I've been finding. So in terms of neuroimaging, there have been a lot of studies, not in different types of drugs. In cocaine in particular, there's not a lot of research, and there's an inconsistency on the results in most of the research. But there's some meta-analysis that can give us a little bit of results that are similar between different studies. In terms of neuroimaging, what I study is first morphology. We do cortical thickness, for those who don't know it. We do more basic morphology of volume-based representation, volume-based analysis, which is here represented, which is analysis of the thickness and the surface area, and volume is just a matter of measuring the voxels. And we also do segmentation of different areas using, I will talk a little bit about the tools we use, but I use basically the ones in McGill, which is CIVET, and there's one called the maggot brain that we use, developed by Malad Chakravarti at the Douglas Hospital in Montreal. And we do the segmentation to also measure, as I said, surface area of the subcortical structures as well, besides volumetric analysis. But we also, sorry, I forgot to say that besides surface area, we can also measure displacement of this area, so the shape can change. So we measure all that. And then we, in studies with cocaine, this is in general in different types of drugs, not only cocaine, but also methamphetamines and other types of drugs. Well, this is a meta-analysis by Escher, and they have found that there's a reduction in volume in cocaine addicts, but also in other types of addicts in these areas particularly. So in this area, like the insula, for example, and the striatum, they're related to cocaine addiction in rats and mice. So most of what we know is from these models. So you have to separate them, because you have to know that we are not rats, but we have similar areas. And there's a negative correlation between the volume of these areas and the years of use of the drug. So they kind of relate to how much time they have used the drug. So it might be a toxic effect. However, no one knows if it's a real toxic effect, or if it's something that beforehand they had in the brain, and then it developed a little bit more with the drug, or if it's an effect of the addiction itself and not any toxic effect. There's also another study by Escher, which are some of the groups that are focusing on this, on morphology, and that the siblings, of people that are unaffected siblings or non-addicted siblings also have this brain changes differently from healthy control. So it might mean that they already have some kind of predisposition to this, either the addiction or the intoxication problems. And you can also, they have also measured this in comparison to like impulsivity. Well, you cannot see it here, but impulsivity, for example, which areas are related, so which morphology problems are related to this, for example, impulsivity traits, and other types of traits like duration of cocaine abuse, for example. So there's a lot to do with, there's changes in the brain that can be related to changes in cognition and the personality, etc. And for example, in this case, in this study, they have found that the thickness of the cingulate cortex actually is different. So normally, when you have the scores of impulsivity in normal controls, it goes like this, and then in cocaine users, it goes like this. So we still don't know what it means, really. But in comparing clinical scores with cortical thickness, it's a bit, difficult to interpret. But there's a connection apparently. And in rats and mice, we have also seen it. This is a study with morphology and age. So there's apparently a change, besides the damage of the using the drug, also apparently people with addiction, and also you can see it in rats and mice, they age, they call it like an accelerated aging. So they have these changes, like increasing volumes in these areas like the orbitofrontal cortex, or reduce a volume in nucleus accumbens with time. You can also see this with the shape analysis, which is what I'm going to talk about a little bit more, which they have changes in the shape of the striatum, for example, and this area is also very related to addiction. So at least in this model we can see it, and in humans also we can see this accelerated aging, you can call it, where you have these individuals that have increased volume as opposed to what you will think. They have increased volume in certain areas that go against what normals, for example, controls are in black. They should go like this with the age. So the more age you have, the less volume in this area. And cocaine addicts have more volume with time. So it could mean that there's some kind of damage, some kind of perturbation there that's provoking that. And in diffusion analysis, there have not been a lot of studies in cocaine addiction. So roughly, there's some studies have shown that in humans, for example, we have FA reduced in cocaine addiction, especially in corpus callosum. Yeah. So, and they have seen this in these areas. And in rats, there's a study where they saw the same effect, which is a reduction of FA in other areas of the corpus callosum, a little bit different. But in this model, it's easier to just dissect the rats and see what's going on. And they have seen that there's a change in the histology there. So the structure of the white matter changes, and that's related to the FA values they have seen. So we can also use BTI to study this population, though the effects are not huge like you would expect. And the other way to study this population is with connectivity, as everyone here probably knows about. So structural connectivity, functional connectivity, and of course effective connectivities. There are some models that we can use to study this population that I want to use. In terms of resting state and functional connectivity, there's a relationship, of course, as well with the addiction. So in these studies with rats and studies with monkeys, there's a consistency there. For example, this is cerebral blood volume, and in rats that are already addicted to cocaine, when they administer amphetamines, their volume goes down, and it depends on how much they have been taking the drug, how much drug they take. And when also in this type of in monkeys, they studied this connectivity between dorsolateral prefrontal cortex and the nucleus accumbens. When they administer cocaine, the connectivity there goes down. Yeah, it almost disappears. So there's a clear effect of the drug and also being addicted to the drug in terms of functional connectivity that we can study in humans as well. And they have done so, but there's a problem. We have the studies that have been investigating this. They have found sometimes contrary effects, like increased connectivity versus decreased connectivity in the same areas in this type of population. There's several studies like this one from Sutherland and WHO, which is basically is the same team at NIH, that they're studying functional connectivity, and mainly the same areas are related, which is the mesolimbic cortical pathway of the dopamine that is affected in this subjects. However, we don't. Sometimes the connectivity, as I said, it's increased. For example, in this case, sometimes it's decreased, and depends on the area that you study. For example, the caudate nucleus here, with the dorsolateral prefrontal cortex, they have seen an increased connectivity in this subject. This is resting state connectivity, as opposed to in the ventral striatum, they have seen a decreased connectivity, for example, with the cingulate cortex. However, I will talk about in the end what our results have been from this study, and we found basically this one, we found the opposite in the same subjects. So it's a problem that it's not very consistent. But you can also relate this connectivity with, for example, how much money they spend in the drug, the impulsivity tests, which are clinical, which is just a questionnaire. But there's a way to relate it. In this case, they found that the dorsal caudate has this relationship. The connectivity between the dorsal caudate and the dorsolateral prefrontal cortex is related to impulsivity traits in this population. So how can we study this? So one way, at least in resting state functional connectivity, we can study it by seed-based analysis and network analysis. The networks that have been known to be affected or that have been studied in this case are the default mode network, salience network, and the executive control network. Those are the main ones that are kind of affected, and seems to be there's a relationship between, for example, impulsivity and salience. And usually you see a decreasing connectivity in the mesocortical limbic pathway. However, sometimes you see this increase in the connectivity. So what does it mean? It means that it's very difficult to study. That's what it means. It means that they're not rats. We cannot control what the person intakes. We can only measure it and ask them. And sometimes they lie. They're people. And they're people taking drugs, so they tend to lie. So it's very difficult to study. So sometimes it can mean that the areas that you're studying and the connectivity between them can be different. Even if you study the same ones, can be different in the same population. So you have to choose your areas very well. The sample could be also a problem. It's not the same to study. And this is also has to do with this. Is it a crack cocaine or is it inhaled cocaine? Apparently crack is worse because it's mixed with other things that are not controlled. I mean, it's not pure like the ones they give to the rats. So they probably get other effects. And this also has to do with polydrug use. That means that no one uses just one type of drug. Not only cocaine, they do cocaine and sometimes alcohol. But the main drug of use is cocaine in their case. So that could also be a problem. So it's important, I think, to relate these variables, these imaging variables, with clinical and cognitive variables to actually find something biological and interesting. Because if you only compare everything to everything, then it loses that meaning. It's not that robust as other type of populations. So I just talked about this. And there are no longitudinal studies in humans. That means that we don't know if these changes have to do with differences in the grouping we're doing, or if it's something longitudinal. It has been done in rats, and it has been shown that there are, this changes actually are very consistent in time, but no human longitudinal studies yet. Actually, there's one that is just starting. I just saw this project in ResearchGate. So there's one starting, but it's no results yet. This is in Switzerland. And no one has studied actually structural connectivity in this population as well. So there's a lot to do here. There's a lot to learn from the murine models. That we can do in the humans, and there's a lot of things that the humans model, you could say model, can do to study the mice. But then, are these changes, the clinical, cognitive, and neuroimaging before and after the drug, and are they related to abstinence? Because a lot of this, especially in the US, the studies are done in people in abstinence. So also we don't know if the changes are just related to the abstinence or not. So I'm just now going to talk about the project that we're doing at the National Institute of Psychiatry. This is the team. I'm not the only one. I'm the only one. I'm in charge of the neuroimaging part in acquisition and all that. And there's a clinical leg by Jorge and a cognitive leg by Ernesto, which are so the cognitive part is not during the scanning. It's aside because we want to see this thing separated. And these are my students that are working on the project. And this project is financed, so we have money for it, and it's been running since 2014. Before me, there was already running, so the sequences were almost ready. So I just tweaked a little bit. And the main questions in the neuroimaging here are, what are the structural characteristics, structural, functional, and connectivity characteristics of the cocaine addiction in our population, and its relation to the clinical and cognitive characteristics. So if there's a relationship between that, and that's very basic, but that's something that hasn't been studied that much as you would think. And also it's a Mexican population, so it's completely different in the sense and how they take the drugs, and which drugs they take. So the basic hypothesis here is that there's a difference between in the imaging, so in the structure of the brain, and also the connectivity between groups, and they will be related to the age, the type of drug, how much they're consuming, and the clinical and cognitive characteristics of the population. And the method, of course, at the moment is just cross-sectional. We still haven't. Well, we need money for the longitudinal, so we're starting with this cross-sectional first, and we're doing, as I said, clinical, cognitive, and neuroimaging acquisition. So the criteria, I will just go quickly, which is just adults, men or women that are addicted to cocaine, right? Could be crack and could be inhaled, but we're separating them. And exclusion criteria as well. Saying this, they have to be at least as healthy as possible, the addicts, and the healthy controls, they have to be also healthy. And this is done with a lot of testing that I will show. So the final sample size will be 100 patients with cocaine addiction and 200 healthy controls paired by so hemispheric laterality, so the Edinburgh test, sex, age roughly, and we're trying to pair for education and socioeconomic status, but it has been proven really hard to find these people that are healthy, because mostly we have low education at high ages. So to find someone at 45 that has not studied only primary school, that is healthy and then wants to participate in the study is quite hard. So we're trying to just use this as a covariate instead. And so what they're doing is we screen them, we recruit them, and they have a first interview, and then they have the clinical leg, the cognitive, and the neuroimaging done during the same week. Usually these two are at this, before this could be this one or this one, and the clinical is before or afterwards. The clinical variables we're measuring are related to the person in general, like the quality of life, dominance, etc. There's an addiction testing with several questionnaires they're standardized about their taking of the drug, the craving of they have, and we have a urine test as well to test if they have taken the drug, because we're measuring people that are not in abstinence mostly, so they should be taking the drug usually. We try not to stop that, but they are in treatment, most of them. And we also have psychiatry batch of questionnaires. Also, this one is very standard, which is the Barrett score of impulsivity, but there are other ones related to their. So this is how we test for any psychiatric disease besides the addiction. And my student Tanya is studying. Is it called the borderline syndrome? So about the cognitive test that we do, we try to measure. Ernesto did a battery of testing that is related to all these tests that are standardized, just to test different aspects of the person. So like executive function, personality, theory of mind, and we have all these tests about working memory, cognitive flexibility, attention, and decision planning. So we have all these tests done in all the persons outside of the scanner. So this is something to take into consideration. And the imaging variables are, well, we have the Philips Ingenia 3 Tesla scanner with a 32 channel coil, and we just do well, this is a scanner. And we have several sequences that we're acquiring. As you can see, the multi-shell DTI that we have, it has a shell of 1000 B value and then a 3000 beta value. All of them have opposite acquisition to have field mapping. And we have the T1, very standard T1. And we have a special sequence which is called the fast kurtosis imaging. I will briefly talk about it. And in functional analysis, functional sequences, we have the resting state fMRI, which is a TR of 2, right? And a 3x3x3 with 300 volumes. So it's 10 minutes of resting state that we do with fixation cross and eyes open. And the analysis plan is very basic. Start with, we will have a lot of data. Well, at the moment, we already have some of it. So the morphological variables will be the cortical thickness, surface area, and curvature of the cortex. We'll have the subcortical analysis using volume and shape, and also surface. And we'll do parcellation with atlas, with different types of atlases to do connectivity. For the white matter, we'll do normal tractography at the beginning. We just want to do the connectome for this. And microstructure will be measured, we'll measure with, I will talk a little bit about the kurtosis method that we're using. And we'll do a connectome type of study. We already have been doing seed-based analysis and ICA. We still have to do the graph theory analysis for this. It's a big sample. And also relate this to all the variables that are not imaging. So this will be the challenge. So we have at the moment 134 participants already scanned. We have eliminated 30 for different reasons. And so at the moment we sit on healthy controls of 48 healthy controls, 56 addicts. Of those, 38 are crack addicts and the other ones are addicted to inhaled cocaine. and they actually differ a lot in the socioeconomical and education. So higher education, more money, and less education, less money. So we divided this in different sub-projects that we call them. So the first one is just a question about the morphology. So are there differences in volume, shape, and the microstructure of the striatum and thalamus that are areas very related to addiction in patients with crack addiction only? And we only studied crack addiction because at the time we only had mostly crack addicts, and also it's the most interesting for me, at least because of the socioeconomic problems they have, and also the cognitive problems they have. So and that these differences relate to age and time of consuming the drug. So we have this. So these are the data. We measured 36 at that time, and 40 controls, and those are the characteristics of the population. And this has been done with a collaboration of the group in McGill with Malar Chakravarti, who's are the ones developing the tools for this. And in the other sequence, the Kurtosis imaging is developed by Brian Hansen and Simon and Sune at Denmark. So firstly, with the morphology study, what we use is, besides CIVET, in this case, in this study, we used what's called the maggot brain. So you can see here the citation, you can look for it. So it's basically a different way of trying to figure out individual segmentation of the different areas of the brain. In this case, I was interested in mostly this, which is the striatum and the thalamus, and also we have subdivisions of each. It's not perfect, but it's one of the best tools I work with, that actually you can segment subnuclei of thalamus and striatum. And my student Pamela is actually working on this too. And we also did diffusion kurtosis. So if you know about diffusion kurtosis, it's that it's a different way of measuring white matter, and that it has more specificity for problems in the structure itself and not the concentration of water. So what have been found is that there's a big relationship between kurtosis imaging and actual damage in several areas of the brain. In this study with a mirror model, they have found, especially in these proteins in the brain, they have found that were related to kurtosis, mean kurtosis differences in this. I think it was rats, if I recall. So it gives you different information that the regular DTI gives you, and you can get the mean kurtosis, for example, from regular DTI. So you don't have to actually acquire anything new, just do different calculations. The thing is, with this group in Denmark, with Brian Hansen, they were trying to develop more clinical type of sequences. So what they did was, it's called a fast kurtosis imaging, and it's basically, they have published it. And it's a way to do faster mean kurtosis for the clinical setting. Especially they have studied it in tumors and stroke. And it just basically takes 2 minutes, so the sequence is quite fast, and it gives you the same results or almost identical results as the mean kurtosis done in a full DTI. So what I did was adapt it to the 3 scanner and the psychiatry department, and what it does, it just has, I didn't put it here, but it's a protocol of actually 1, 9, and 9 directions. So it's a multi-shell they have, and it runs really quickly. And it gives you mean kurtosis, which is what we're trying to figure out, to find. So the analysis were very basic. So what we did was just compare between groups and then covariates. And so we have published this as a preprint, and it's now under review and translational psychiatry. So I will show you a little bit about the results we have at the moment. So we found that, as opposed to other studies, that sometimes they find increase, as I said, sometimes they find increasing or decreasing in the volume. We found no differences in volume and kurtosis between groups. So if using the covariates that we use as in age, for example, we didn't find any differences in between them. You can see here the mean volume of all of them, and striatum and thalamus, and we didn't find anything between them. So there was no difference between groups. However, there was a difference between the striatum shape, but not in the thalamus. Here you can see as it's in the ventral striatum, which is the nucleus accumbens, an area very related to addiction, we found what they call an inversion of, or a difference in the shape. So it's inward displacement, or we call it, in the cocaine addicts compared to the controls. And this was not significantly related, but it was interesting to see that there was a relationship, an interaction between age. So this is the control group. With the age, the inward displacement tended to go down, and with the cocaine addicts, it tended to go up. However, this was not significant at this point. And there was no correlation between the age and consumption and the shape of the nucleus accumbens. So this one was not related to the years of use, surprisingly. But there was a significant interaction between the volume of the striatum, especially precisely in the post-commissural caudate with age, but not in the thalamus. And you can see here, these are also the main results that we have that relate to this aging or quick aging that we talked about, which is in the total striatum and especially the caudate nucleus. We found that the healthy controls reduced, with time. These are the healthy controls. Reduce the volume with age, but especially here, the cocaine addicts had an increase in this, and the effect wasn't very high. Also, as I said, the effects were really, really low, which I was surprised to see. But this is very consistent with everything that we have found in the literature. And the nucleus accumbens volume did not have an effect there. We also found a correlation between a part of the thalamus and years of consumption. There was a correlation there at our 0.52. And we found that although there were no difference between the mean kurtosis using the other sequence, we did find the same age interaction, so group age interaction that we have seen here, especially here in the thalamus, but also in the striatum. So in both, we found this different, and the effect was a bit higher than this is effect size. So this is the effect was a bit higher in than with the volume. So in conclusion, what we found was that volume did not have an effect in our data, and it was higher in the striatum, just higher but not significantly higher in healthy controls than cocaine addicts. Sorry, the other way around. And the difference in shape was in the nucleus accumbens, which was surprising. No one had done a shape analysis before in this type of population, so we were surprised to find it exactly there. But then it was a bit tricky that we didn't find this interaction with the age. This correlation with the age of use of the drug, with the years of use. Sorry. So there were interaction with age, as we have seen before. There were no differences between groups in volume and kurtosis, mean kurtosis, but we did find this interaction with the age, contrary to observe in the normal population, and we corroborate this with other studies. They find that volume reduces with age, and we found an increase in volume. And so mean kurtosis, the fast kurtosis sequence hasn't been, it wasn't used before for addiction. So we found some good result with this, and I think it's a good type of sequence to put in vivo studies at the moment, and we're going to keep studying it in comparison to the mean kurtosis found with DTI. And again, I think volume is just one measure that is used a lot, but I think it's incomplete. I think you need to use more than one type of a measurement to actually find something important there. So in comparison only between groups is not enough. I think you need to relate to something about the addiction, about the subjects. And of course, the limitation we had, the kurtosis sample was quite small because we started measuring this after some time. And nicotine dependency could be a good covariate, but we didn't find this relationship. And polysubstance use, as I said, it's a problem, and it's a huge problem in crack cocaine use because they use other types of drugs as well. And we couldn't compare the type of drugs. So the next thing to do in this case is, now that we have more sample, when we finish the whole sample size, to compare between different types of, as I said, inhaled cocaine and crack cocaine. As a second project at the moment, where Luis is also involved, and Sarah Ellen, probably you know these guys, and my students, they're working on inhibitory control. So they're interested in, there haven't been a lot of studies in inhibitory control in humans. So we're trying to figure out if there's a connection between the problems in inhibitory control and memory that we're measuring outside of the scanner and the resting state connectivity, if we can predict that they will have these problems, and if there's a difference actually between healthy controls or not. And so this is the hypothesis. Basically, what I said is that there will be a relationship there between the cognitive and the functional connectivity. So what we did was I'm just putting here the test for the inhibitory control with the flanker effect. I don't know if you don't know it, but it's just a test of inhibitory control, and the go-no-go test, which is stop signal. And then we have a perception of impulsivity with the Barrett. This is the questionnaire that you ask people about how impulsive they think they are, which is a bit different than actually doing a task. So we have both. And usually what they do in studies is just this one, the questionnaire, which I don't like that much, which is why I prefer to also include these ones. And we have working memory. We have the GSPAN and letters and numbers, and the Brown-Pearson test, which is a stress test. So we have different types of testing, these domains. And we're going to do structural connectivity, functional connectivity, and affective connectivity in the end. We haven't finished it, so this is very preliminary. What we are studying here is first the seed-based analysis using FSL and AFNI, with in mind using this ROIs that relate to the mesocortical limbic system, the pathway. The networks what we're studying are the default mode, the salience network, and the executive control network. We're going to do graph theory using the brain connectivity toolbox. And we're doing as I said, a multi-shell GTI. We're trying to, want to use MR-Trix because Luis is working with this as well. And we want to see the difference also between drug type, not only between groups, and the relationship between the cognitive variables. So these are roughly the areas. They're not the ROIs that we're using, it's just so you can see them. And of course the hippocampus and medial frontal cortex for the memory, for the working memory. So far this is very preliminary. This is for posters they did. And so we found that, yes, the cocaine addicts perceive themselves as more impulsive. The flanker effect and go-no-go had no differences between groups, and this is not surprising at all because there are maybe one or two studies that have found these differences, and the norm is to not find these differences. So apparently this type of, so the addicts actually function the same, and they're functional addicts. They're not addicts that are in an institution. They're people, like normal people. So they're normal people that actually just take the drug. So we didn't find any difference there. But we did find a difference in the reaction time. So they were equally good to answer this flanker effect and the commissions, the go-no-go, but they were slower. And this is slower in terms of cognition. So it was just milliseconds. It wasn't that long. And you can see that the bars are really high because this is only between, it's not between the drugs still. It's just between healthy controls and the cocaine addicts. But still there's a difference there that we'll try to find with more subjects. But it's very subtle. It's not a huge effect. But there are differences in the reaction time. So they're slower, but they answer correctly. So it might be that there's a problem there in the connectivity between in these areas related to that task. And the years of consumption of the drug were related to the flanker effect, and the reaction time. So there was a relationship between the drug use, the amount of drug they use, and how they answered. And so far we have only studied the ventral striatum and the superior part, this is a map. You can see the connectivity, the functional connectivity map of the healthy controls and the addictive to cocaine. They look roughly the same, but they have these differences. And so when we see this contrast, we see these differences in so it's an increased connectivity in the right cingulate cortex and the right caudate and this area, which is near the inferior frontal gyrus, which actually, well, you can see the areas here. And this increasing connectivity was related to the Barrett test, but not to the cognitive test. So we're still trying to figure that out. And this is very preliminary, as I said. But this result is very consistent with what we've seen with the studies from the NIH. However, what we found was for example, here in the right cingulate cortex, and in this area connectivity, we found an increased connectivity, and they found a decreased connectivity. And I put it in the beginning. So we still need to figure that out, but it seems that in our subjects we found an increased connectivity, which could mean many things. And here are the maps of the different networks that we found. We just mapped them at the moment, and this is the only picture we had at the moment from one patient. Now we have another one thanks to Donald, but this was done also using MR tricks, so you can see that we still need to process all that. As a third project, we can call it a third project. My student, Tanya, sorry about that, she's interested in as I said, in borderline syndrome. And these patients have, with borderline syndrome, most of them are actually cocaine addicts. So it's interesting that she wants to study that. So for this, she wants to study something called dual pathology, which with people that have cocaine dependency or cocaine addiction, borderline syndrome, with and without a cocaine addiction. So we have the sample, the end sample will be, I forgot 20 per group. So it will be cocaine addicts with the borderline syndrome, cocaine addicts without borderline syndrome, without any addiction, and healthy controls. And she's basically she's doing her master thesis about this. So she's going to only study at the moment just the FOMO network. But the idea is to continue this a little bit more, because it's an interesting area. And we're also starting this clinical trial that we're interested in seeing if TMS it's actually reducing the addiction, because some studies suggest it, but no one has done a long-term study. So we're doing this, it's already funded, and we're going to measure, we're going to stimulate them, to stimulate the patients, and measure here it should be at 2 weeks, 3 months, 6 months, and 12 months. We're going to measure MRI, the same sequences that I've shown you. We're going to measure them in time for a year. That's the goal. And the idea is to first test if this works. Second, if it works, why does it work? And if it doesn't work, why doesn't it work? And also, I'm interested in we're going to stimulate the dorsolateral prefrontal cortex, because it's the normal one that usually you stimulate in cocaine addiction. There are maybe like 3-5 studies about that, but there's no real reason, only that it has been used in depression and that it reduces craving for 8 hours, but no one has done more than 8 hours. So I'm going to try to analyze. So the cortex. The idea to use the cross-sectional sample that we have to study connectivity is to find the connectivity of the areas related to the drug use, which is usually the striatum, and see which areas of the striatum actually connect to the cortex functionally and structurally, and try to find a different target area to simulate in this patient. So we have 2 groups, one that is going to be simulated in the dorsolateral prefrontal cortex, and another one that will be stimulated in a new area. If by 2 weeks we don't find anything, that we'll discard this, but hopefully we'll find that there's some area that is maybe better or different and the way we will know if it's better or not is if they become less addicted to the drug, if they don't use it anymore, and if they get their cognitive function better. And maybe we'll find that even they keep taking the drug and TMS makes them better in the cognitive, maybe not, we will know. But the idea is to relate this clinical and cognitive improvement or detrimental detriment with what we find in functional connectivity and structural connectivity. That's the main idea of this study. And we haven't started it. It's just got the ethical approval. We're not right now uploading it to clinical trials. Hopefully we'll start in November, December. So, and just to finish, we have this collaboration with University of Oxford and ours, where Enrique, who's a postdoc there, he's an engineer, and he has this model it's similar to machine learning, but they actually have this pattern functional model, pattern search model, where they constrain the functional connectivity by the structural connectivity. So they're basically going to grab our data and analyze everything. So the clinical leg the cognitive variables and connectivity variables and try to find patterns there that are related to function. This is what we're working on at the moment. It's starting, and also, as I said, I'm starting with rats as well, and we're going to study with Luis and my colleague Alberto, who's at the University of Nuevo León. He's working on a model of addiction to hypercaloric diet, which is fats and sugar. So the rats that he's using actually get addicted to this food. So the model it works at the moment, and well, you can say their children actually have cognitive problems and behavioral problems, and actually some of them are also addicted to hypercaloric food. So we're going to study at the moment only the macroscopic imaging, which is using volume, just regular T2, T1, for the moment, and see if that relates to changes in the behavior and also microstructural changes. He is going to do some immunohistochemistry and histology works besides the imaging. And the idea is to continue this a little bit more, but this is the first study we're doing in the scanner with at the Institute of Neurobiology. And that's it. I'm finished with talking. Thank you. So, any questions?